Hi, my name's Nate Gearin, and I'm excited to be with you to share my project, my final project for the Bioinformatics Algorithms course, in which I created a phylogenetic tree for the superorder Lauraceatheria, which is a group of placental mammals. So, I've always been interested in history, and one of the downsides of history by itself written history is that it only really goes back 3,000 years at the most. And so to me, when I learned about the ability to infer evolution through analysis of DNA sequences, which gives some glimpse into history that occurred, from tens of millions to hundreds of millions of years ago, that seemed like a really interesting topic to look into. It also allows us to compare species in terms of similarity and infer ancestor species. And where some methods of phylogenetic tree inference allow us to hypothesize about how long ago species speciated, how long ago they became distinct species. Even though I'm going over this in the context of bioinformatics, phylogenies and more generally the idea of a tree of life is not a new concept. Here there is a image called the Generelle Morphologie der Organismen, a general morphology of organisms, that was published in a book in 1866 by Ernst Haeckel. And that is basically concurrent with when Darwin was sharing his ideas about his theory of evolution. So these types of trees of life have been around for at least 150 years. But over this time, they're definitely changing. Here's an image of a more recent tree of life, one that has thousands of species and different groups of species as well, that was published just a few months ago in Nature. And they used an interesting approach to create this tree of life, which is different from the approach that I took. They used whole genome sequencing called metagenomics, and they were able to access groups of species that had never been sequenced before, which is why their eukaryotes seem like such a small part of this tree of life. Over history, there have been a couple different ways to create the trees of life. There are character-based phylogenies. So, for example, does a species have a cleave and hoof? Or what does its bone structure look like in comparison to other species? More recently, with the discovery of DNA and the ability to sequence genes, there have been phylogenies created on sequences. And this last one, which we just talked about, was whole genome sequence comparison. In general, the method that I took had these 6 steps. First I needed to identify a set of species that I was interested in and that shared a similar gene. Then to obtain the sequences of the gene, which I did through Ensemble. Then to align the sequences, calculate the distance between the sequences, use the distances to infer a tree, and then to actually draw the tree. Now, I did these 4 steps of these 6 steps. There are many tools that are available to help infer phylogenies. So for the 2 steps that are gray, I used widely available tools that I'll demonstrate for these steps. The species that I chose were the common species that every 4-year-old is aware of, and they include the horse, the cat, the dog, the panda, alpaca, the pig, dolphin, cow, and sheep. These are all members of the Lauraceatheria superorder. And as an outgroup, which allows one to root a tree, I chose the two species, chimpanzee and humans. The gene that I chose to use as the comparison point is the 16S ribosomal RNA, which is found in the mitochondria in cells. And the ribosomal RNA has been termed the backbone of a universal tree of life. And the reason for that is that it is a very conservative gene that is shared among all species, all eukaryotes. So I now want to demo how one goes about finding these gene sequences. I'm going to go to the Ensemble genome browser, and since the humans have this gene, I'm going to go ahead and click on human and look at the mitochondrial chromosome MT. Now I happen to know that this particular sequence is located in the beginning of the short non-coding gene sequence. So I'm going to jump to this region view, and we see here MTRNR2. That's the sequence I'm interested in, the 16S rRNA. When you click on it in the region view, you can then go take a look just at this gene. And Ensemble has this really nice feature of comparative genomics. And I'm going to go there and click on genomic alignments, which is just a shortcut I used to get this same gene in the different species I was interested in. Right now, Ensemble is finding those sequences and actually generating its own phylogenetic tree, which is here. And here are the sequences that it provides for a bunch of different species, and I was able to find all of the species I was interested in this group of species. So the next thing I want to talk about is aligning the sequences. There are many tools that exist to aid with multiple alignment. I chose what I think is the most popular one. I called a Clustal Omega, which is the most recent version of the Clustal software. And the European Bioinformatics Institute has an online version of the multiple sequence alignment program from Clustal Omega, which I'm now going to show you. So first, I obtained all of the individual sequences. It is then necessary to align the sequences. I'm sorry, not to align them, to put them into a single FASTA file, which I did here. You can see here's the cow sequence, the dog sequence, horse, etc. I'm now going to upload this into the Clustal Omega program. I'm going to choose my output format as FASTA and submit the job. Now the job just takes maybe 15 seconds to do, but I already have a version that has returned. So here's the multiple sequence alignment. I'm going to go ahead and download it. Let's call it species align.fasta. Good. Next, I want to show you my program that I wrote that creates a distance matrix and then uses UPGMA to create a tree. So first, the distance matrix. So there are 11 species. Let's take a closer look at these results. There are 11 species, and these numbers represent the distance in between each of the species. It's in a matrix format. So the distance between the human and the chimp is 0.055153, and this is done for every pair of species. The format that I chose here is a format that can be read by the Philips Bioinformatics program, which is a existing program that can draw trees. And when I was developing the program, I used Philips as sort of a baseline test to see how close I was getting to what at least Philips thinks the right results are. So when I did this distance matrix, I then would feed it into Philips to see the tree it generated and make sure everything was sane. So this distance matrix that came from the multiple sequence alignment, I used a approach that's called the unweighted pair group with arithmetic mean, UPGMA for short. But there are different approaches to clustering. I think we've covered both the UPGMA and neighbor joining in the course, but these algorithms have different assumptions on what's termed the molecular clock, or how fast mutations occur in a gene, and if they occur steadily, or if there are periods of rapid mutations and then periods of relatively slower mutations. These different algorithms also have implications as to whether or not the tree you create has a root. UPGMA does allow you to have a rooted tree, and you can also include different concepts of models of evolution. The one that I used is a simpler one. It's called the Jukes-Cantor evolutionary model, and basically it assumes that a gene, the likelihood of any base in a sequence to change is equal for all bases, and that the probability that it will change again is also equal. So the outcome of such an evolutionary model is that the distances that I created in the distance matrix are slightly larger than the actual distances in between the sequences, because it's accounting for cases where a base has mutated and then mutated back to its original base, as well as other changes that are not visible from the modern sequences but might be in the history of a sequence. So you've already seen the construction of the matrix. Now let's take a look at how I inferred the tree using the UPGMA algorithm. Okay. So that had a lot of output. Let's take a look closer at what is here. What you see here is the state of the distance matrix in each step of the algorithm. After each step, two of the nodes are joined, and these two nodes that are joined are the ones that are closest to each other. So in step 1, there are only the leaves of the tree are available, but then, for example, the two species that are most closely related to each other out of all of these 11 species are the human and the chimp. So step 2 joins human and chimp together to make an internal node. Step 3 joins cow and sheep, because they're the next most closely related species, to make a second internal node, and so on, until there are only two species left. And finally, I print out a dictionary of all of the different internal nodes, which allows me to then go and draw a tree. Oops. And this is what it looks like. So here you see that here was the result of the UPGMA algorithm, and here is the phylogenetic tree. We see that the cat is most closely related to the dog, which then had a common ancestor with the panda, which had the common ancestor with the horse. And likewise, the dolphin and pig, as well as the cow and sheep, have a hypothesized common ancestor, which then have a hypothesized common ancestor with each other, and then with the alpaca. Here's the outgroup of chimp and human, and our common ancestor with all of these other animals. So one of the things that I found as I was creating this distance matrix is that perhaps the gene that I chose is not the most optimal gene for species that are so closely related. The 16S rRNA gene is fantastic for when you want to infer deep relationships, but for because it's such a conservative gene, it doesn't mutate very quickly, and which means that the distances between two species that are different, like chimp and human, are very small. Secondly, I found that as I was testing my algorithms against the Phillips program, that Phillips has an option for randomizing the tie-breaking part of the tree creation, and I found that this could lead to a different phylogenetic tree. So for example, here I have the cat and dog as the most closely related species in this clade, but if you and to say also Phillips does too when it doesn't randomize the input, but when it does randomize the input, it finds that the dog and the panda are actually more closely related and then have a common species, a common ancestor with the cat. So how does this phylogenetic tree compare to other phylogenetic trees that are published? Well, the contours are largely similar to the ones that are provided by Ensemble and Orthomem, but there are slight differences. I account for these slight differences because both Orthomem and Ensemble are using, they don't just do one gene differences. Ensemble by default compares a much larger sequence than just one gene, and Orthomem compares many related sequences from each species to each other species. And that is the end of my presentation. So I hope I haven't lost you and that you enjoyed it. I have a bunch of references here that are very small, but if you're interested, I'll post my PowerPoint and make that available for you. So thanks for your attention.